I discuss on the fresh topic called as uh, docking using a software called MZ and now I'll explain you about the docking software, which is very user friendly, and how to dock. I'll just show you. So this is a software called MZ Dock. To dock using this software, you need to just have to a single software will give you multiple answers. You just have to click on the MZ doc available on the Google, that is on the source page or on the GitHub, and download it. And it takes very optimal or minimum space. And once you download it, you just have to uh, fetch it on your computer in this fashion. So first and foremost thing is you need to know that MZDoc operates for the Autodoc 4, where the command prompts of the Autodoc Pro are automatically run by the software, where you need not do anything. Similarly, the ligand preparation, protein preparation, binding site selection, docking, analysis, everything is done by the software where you need not do anything. Even if you follow the step-by-step procedure, you will understand what exactly the docking is in a simple and foremost way. Let us start and explore what exactly is happening. Click on the new and it will start your the docking, and it shows that from where you have to cite this software when you use it in your research. And this part tells me about selection of ligand. Either you can load, click on the load, you can in these many fashions. So I'm just selecting the PDB part, and I'll just go on to desktop and I'll check any this first molecule I have in PDB. I'll just click OK. And you have to know that these force fields, there are multiple force fields available in this software. This is integrated with this software. There is MMF 94, 94S, GAF, chemical, and UFF. These softwares are the force field which are useful in energy minimization of your molecules. If your molecule is a simple and small molecule which is less than 500 kilodaltons, as well as if the molecule is having, what happens if your weight is very less and it is a very small molecule, you can use MFF94. Suppose if it has some heteroatoms, it has some chirality, if it has some enantiomeric or diastereomeric parts, then you better go for a force field, that is GAFF. These are all the force field which creates, and what you say, smallest energy conformer or perturbations and iterations to prepare the ligand. Just click on prepare and see what is happening in the background. The background is present on the left side of the screen, which shows how the molecule is prepared, how many iterations are done, what is the total time consumed, and everything is displayed in the command prompt window of the MZDoc. Just you have to click OK. Once you click, the ligands are prepared for dock and it is ready for docking. Now you have to select protein. This protein may be either you load it on your from your computer or you fetch it from the protein data bank or alpha fold. Let us fetch it from the alpha fold. Just click extract. Once you extract, this protein will directly fetch onto the protein data bank. So now once I have clicked, the protein is already fetched onto the protein. Now it says if it is an uh, once I click onto the any chain, because it is having two chains, you have to only click one chain. And if you, as the number of chains increase, the complexity of the protein increases. If it is a single chain protein, then is no problem. And if it is a multiple chain, or you can say you can select only one amount for docking, and you just click the confirm on this chain is extracted. Now this is the most important part of the docking, where you have to select the co-crystallized ligand or the binding pocket, and you are to careful in non-selecting those het atoms which are cofactors for docking. In this, there are three heteroatoms. In this 181 is your co-crystallized ligand, whereas sulfate and other heme, that is heteroatom, is for the cofactors. Now to see, and this once you click check, what happens, it's it is specifying that there is no water molecule within 5.2 angstrom. That means within the binding pocket there is no water molecule, so you need not retain any water molecule. Suppose if there are any water molecules within the binding pocket, you are supposed to select those water molecule and retain them, and what is the displacement energy used in doing so, that is also crucial in docking. And just click confirm, and now your protein is prepared. And it is now, once you click on the protein preparation, and what are all the parameters which you incorporate in your autodoc 4 will display here. So these are all the parameters which you incorporate in autodoc 4. Once you click confirm, your protein will be readily prepared, and it is protein preparation is complete. Now once your protein preparation is complete, you have to select the binding pocket. Just click onto the binding pocket. This is the one ATM which I have already showed. This is the co-crystallized ligand for this protein. You can simply click confirm, or if you don't want to use this as a binding pocket, you can go for blind dock, that is complete protein is used as docking, or you can manually select those residues which are used for docking, or you can go for flex dock, that is flexible docking. Just I'm just clicking the confirm here, so you can choose multiple options in your docking pattern. Now just click on prepare. Now your binding site is ready. Now your ligand is ready, protein is ready, binding pocket is ready. You just have to run docking. Now this run docking I have given, and how it is docking, how many simulations are used, how many modes of docking, what is the scoring function, everything is readily displayed on the software itself. You need not do anything. And it also performs the RMSD of the docking. See, the RMSD of the docking is also being performed, how the co-crystal ligand is redocked onto the software, and its results will also be shown here. So this is the beauty of the software. So now it shows the docking is successful. Now just you have to click OK, and it shows that only one pose, that is the ligand pose 1, is successful, and the remaining all 8 iterations have been failed. Now you just click OK, and now it is showing which folder you have to save. So now what I'll do, I'll just create a folder to explain you. I'll name it as YouTube, and I'll just click, and now I'll select this folder, and the results are saved. Now I'll just click on the analyze. Now the analysis is running in this background, and it will show how exactly the protein or complex. Now I have to go on to the same folder, and I have to click, and I have to show the where I have to go for. Just I will click onto the uh, output, and I'll just click open. Now these many poses are there, but we know only the ligand pose 1 is fit for the docking, whereas other poses failed in docking. You just click open. Now what happens, its background is running, and it's showing how the complex is uh, prepared, and complex is being saved in the folder. Now your folder is already analyzed, and again you'll go out onto the same folder, that is YouTube, and you select the folder, and the results are saved, and you just click get protein, again go back onto the same folder, select folder, and your protein is saved. Now this completes your docking. You just close the software, where you need not do anything else. Just open the folder and see what else are available on the screen. So these many information is available here. So this is the, what do you say, RMSD part. This is the output. This is the image of your ligand protein complex, how your ligand protein is interacting with your residues, and this is your complex, this is your complex, this is the RMSD report, These are all the output files which are readily available in your software. You just have to go to the third party software, that is Discovery Studio, and just open on, sorry, let me close all these which I have been performing in my last research. So I am stopping all these things. I'll just present you the current uh, data. Just I'll click on open, and I'll go to the folder which I have already prepared, and simply I'll click on complex protonated, and I'll just click OK. Now this shows me the protein which is used in docking, and similarly I'll go with again you folder, and where again I'll click on the complex. It shows me again the protein with docking, and so these are the fundamentals in docking. Again you will go with complex protein, uh, and or else you have a standard out, and you can directly open this in the Autodoc and see the results. And you can let us see the results in different, what do you see, just go with this part, and you have an output in this fashion of the your protein preparation, that is Ramchandran plot, how your protein is optimally prepared. And you can simply go with this complex which shows how the ligand is optimally fit onto the binding pocket. And you just click on the ligand, define ligand, click ligand interactions, go for show 2D diagram. It will show how it is interacting, and go for ligand non-bond monitor and go for non-bonding interactions, which will clearly explain what type of interaction your molecule is having, whether it is favorable, whether it is unfavorable, what are the residue number you can see here. So what type of interaction this is having, non-bond, electrostatic, hydrogen bonding. So these are all things are possible in a single software, where you have to get and do step by step clicking on a single software. That is what is your talking. So with this I am stopping here.